how do you measure something you literally can't see? I know, it sounds kind of like a magic trick, right? But it's something scientists do every single day. And their secret weapon? It's light. Yep, just plain old light. Today, we're going to pull back the curtain on this incredible technique called spectrophotometry.
All right, so let's just jump right in with a little thought experiment. Picture this. You're in a lab, and you've got this beaker full of liquid that is perfectly clear. I mean, it looks just like water. But you know there's something important dissolved in there. Maybe it's a protein, maybe it's a new drug you're testing. The huge problem is you have no idea how much of it is in there.
So, how do you measure it? I mean, it's completely invisible. You can't just put it on a scale, you can't filter it out. This right here, this is the exact puzzle that spectrophotometry was invented to solve.
And the key to solving this invisible puzzle, believe it or not, is light itself. We're going to use light as our detective, our main clue to figure out what's hiding in that liquid. You've definitely seen this before, right? A simple little prism takes a beam of white light and, poof, it splits it into a gorgeous rainbow. Well, each of those colors is just light traveling at a slightly different wavelength. And being able to break light apart like this, that's step one of our investigation.
But get this, that beautiful rainbow we can see, it's just a teeny tiny slice of all the light that's actually out there. The full electromagnetic spectrum has everything from radio waves to X-rays and gamma rays. And what's really cool is that spectrophotometry can use parts of this spectrum we can't even see, like ultraviolet light, to do its work.
Okay, now here is the absolute key to everything. When light shines through a substance, the molecules in that substance will literally eat up or absorb very specific wavelengths of that light. And if we track which colors get absorbed, we can create a special kind of chart called an absorbance spectrum.
And this is where it gets really powerful. Every single molecule has its own unique absorbance spectrum. It's like a molecular fingerprint. No two are exactly alike. For instance, think about a plant leaf. The chlorophyll in it is great at absorbing blue and red light, but it pretty much ignores green light, so it reflects it, and that's the color we see. This unique fingerprint is exactly how we can identify what's in our sample.
All right, so we've got the theory down, but how do we actually put it into practice? Well, for that, we need a machine called a spectrophotometer. Let's take a peek under the hood. Now, I know this diagram might look a little complicated, but don't worry. All it is a super controlled path for a single beam of light. We're just going to follow that beam on its journey, from start to finish.
This whole journey really just breaks down into 4 simple stages. First, we make some light. Second, we pick the exact color we're interested in. Third, we shine it through our sample. And fourth, we measure what comes out the other side. That's it.
So, let's trace that path. It all starts with the light source. That light then hits something called a monochromator, which is basically a fancy prism that lets us pick just one single color. That pure, single color beam then goes right through our sample, which is sitting in this little transparent box called a cuvette. And whatever light doesn't get soaked up by the molecules in there keeps going until it hits the detector at the end. That detector then spits out our magic number, the absorbance.
Okay, so the machine gives us a number for absorbance. Great. But what does that number actually mean? How do we get from that number to the answer we really want, the concentration? Well, it turns out there's a surprisingly simple and elegant rule that connects them.
Just think about it. It's actually super intuitive. Imagine you've got a glass of really weak iced tea. It's pale, right? Tons of light can pass right through it. So that's low concentration and a low absorbance reading. Now, picture a mug of super strong, dark roast coffee. Not much light is getting through that. That's high concentration and a high absorbance. See? It's a direct relationship.
And this simple idea, the whole coffee and tea thing, is actually formalized in a scientific law. It's called the Beer-Lambert law. And yeah, the formula looks a little technical, but all it really is the math version of our coffee example. It's the equation that directly connects the absorbance we measure to the concentration we want to find.
Now, you don't need to memorize all these letters. The only thing you really need to know is that A for absorbance is directly proportional to C for concentration. That means if you double the amount of stuff in your sample, you double the absorbance reading. It's that simple. And because of this, scientists came up with a clever shortcut so they don't have to solve this equation every single time.
Okay, we have all the pieces of the puzzle now. We understand the principle, we've seen inside the machine, and we get the basic law. So let's put it all together and finally figure out the concentration of our invisible substance.
The trick scientists use is to make a kind of cheat sheet. It's called a standard curve. Here's how it works. Before you even touch your unknown sample, you first make a few samples where you know the exact concentration.
Then you measure the absorbance for each of those known samples and just plot the results on a graph.
And this is what you get. It's super simple. You've got the concentrations you already know along the bottom and the absorbance the machine measured up the side. If you did everything right, those dots should form a perfect straight line. This line, this is now our roadmap. It's our key to finding any unknown concentration.
So here's how you use it. It's almost laughably easy. First, you take your mystery sample and pop it in the machine. It spits out an absorbance number. Let's say it reads 0.4, okay? So you find 0.4 on the vertical axis. Then you trace a straight line across until you hit your standard curved line. And finally, from that point, you just drop a line straight down to the bottom axis. Whatever number it lands on, that's it. That's the concentration of your unknown. Simple, elegant, and incredibly powerful.
And really, that's the whole magic trick right there. The entire technique comes down to this one beautiful, simple idea. By measuring the amount of light that disappears, you can figure out exactly what's there. And this isn't just some obscure lab trick. This is used everywhere, from testing for pollutants in our water to measuring glucose levels in a blood sample. It makes you wonder, doesn't it? If every single molecule interacts with light in its own unique way, what other invisible secrets are just hiding in plain sight, waiting for us to shine a light on them?